A very good morning to everyone. You all are welcomed to cherish and celebrate the 81st Foundation Day of CSIR, the Council of Scientific and Indian Research. With dynamic network of 37 national laboratories, 39 outreach centers, and 3 innovation complexes, CSIR has pursued cutting-edge science and advanced knowledge frontiers. CSIR is ranked 37th among 1,587 government institutions worldwide and is the only Indian organization among the top 100 global government institutions, according to Chicago Institution Ranking World Report 2021. CSIR also holds the 7th rank in Asia and leads the country at the first position. We shall feel honored to be a part of such organization. Now, on behalf of everyone present here, I would like to invite the Honorable Director CDRI, Dr. Radha Rangarajan, and the Chairperson of the Organizing Committee, Dr. Smriti, to please come on the stage and welcome our Chief Guest, Dr. G.P. Talwar. मैं भूल ही गई कि ये भी है. I would now request our Director, ma'am, to kindly say a few words to the gathering for this occasion.
Professor Talwar, Dr. Nityanand, Dr. V.P. Kamboj, distinguished guests, dear colleagues, students, press and media, former staff members of CDRI, and everyone in the audience, good morning to all of you. Thank you all for joining us today to celebrate the 81st Foundation Day of the Council of Scientific and Industrial Research, CSIR, as we fondly call it. A warm welcome to Professor G.P. Talwar, Director Research, Talwar Research Foundation, New Delhi, for so generously acceding to our request to grace this occasion as our chief guest. Professor Talwar, of course, is very well known. He's the founder director of the National Institute of Immunology, New Delhi, and has been professor of eminence at the International Center of Genetic Engineering and Biotechnology. He has made seminal contributions in the field of immunology and medicine, including a vaccine for leprosy, which is approved both by the DCGI and the US FDA. He's an inspiration to all of us, and we are fortunate to have him in our midst today. Thank you. I also would like to welcome his family members who have joined us for this event. I extend a special welcome to Dr. Nityanand, who is, of course, well known and has so gracefully agreed to be here amongst us, and also to Dr. V.P. Kamboj, former director. So both former directors are stalwarts. It is amazing to be in their presence. A lot of history at CDRI was built on their shoulders, and I'm really grateful that they came here today to be with us. Today's event is a celebration of the vibrant and rich R&D ecosystem of CSIR. As we all know, CSIR supports research and development across a broad range of fields, from basic biology to materials science, from food to leather, roads to aviation. CSIR touches upon almost every area of science and technology. Established in September 1942 as an autonomous body, CSIR has a network of 37 national laboratories, 39 outreach centers, 3 innovation complexes, and more than 10,000 personnel who are engaged in scientific activities. Its achievements are many, including the recent development of biojet fuel from non-edible oil, a safe and effective contraceptive, devices to detect adulteration of milk, electromagnetic and surface magnetic techniques for aquifer mapping, and many, many more. In fact, CSIR has granted 90% of the US patents granted to any publicly funded R&D organization in India and has emerged as the largest R&D organization in the country. Dr. Shanti Swarup Bhatnagar, the first DG of CSIR, said in 1947, "The scope of work in each laboratory could perhaps be best described to be the form of a continuous spectrum, at one end of which research of the purest academic type and of the highest quality is carried out, and at the other, the technical development of processes and equipment proceeds by stages." In 2007, the then Prime Minister, in his address to CSIR upon completion of 65 years, said that the mission of CSIR needs to evolve with the times. CSIR's renewed mission, he said, should be to build a new CSIR that will fulfill the aspirations of modern India. In keeping with this sentiment, the current vision of CSIR is to pursue science which strives for global impact, technology that enables innovation-driven industry and nurtures transdisciplinary leadership, thereby catalyzing inclusive economic development for the people of India. More recently, the Director General, Dr. Kalai Selvi, reiterated that all CSIR organizations need to work closely with industry, so our work remains relevant and impactful. CDRI, our own beloved organization that we all have the privilege to be a part of, has participated in the country's development through contributions in new drug development, innovations in process chemistry, and technology development. The work we do here is complex, risky, and highly regulated. This means that taking products to market is more challenging. Failure is not uncommon. CDRI's seminal contribution in developing a safe contraceptive was a big win, a jewel in the crown. We have had other successes, including the anti-malarial Artheta, a herbal formulation for bone health, and others. How can we make success repeatable and impactful? Focused research driven by unmet need and a keen understanding of commercial opportunity, clear goals and go-no-go criteria for progression of compounds, unambiguous clinical endpoints, and a comprehensive understanding of regulatory requirements are essential elements of success. Then we need a problem-solving mindset, hard work, and perseverance, and some luck stirred into the pot. Easy, right? I have confidence that it can be done, bit by bit, one step at a time. As Vincent van Gogh once said, great things are done by a series of small things brought together. Of course, there will be hurdles, both monetary and technical. At each turn, we must look for allies and partners, overcome barriers, be strategic in our thinking, and consistently aim for global standards. If I have to point to one determinant of success, I will say that it is the quality of the teams. There are many examples from history of teams who have won against great odds. The Indian cricket team's historic win at the World Cup in 1983 and the Mangalyaan mission stand out for me as two shining examples of what people can do when they come together with single-minded focus and a clear eye on the prize. No distractions. These examples are an inspiration for us to write our own story at CDRI and take this organization to new heights. As Helen Keller once said, Alone, we can do so little. Together, we can do so much. Guided by common goals for the institution, I'm confident that we can deliver drugs to the market expeditiously and touch the lives of millions of people. Dr. Talwar is going to talk about mission mode research to develop products. I look forward to his insights on this topic. Before I end, I'd like to thank the organizing committee for putting together this event. It involved a lot of hard work and coordination, and I'd like to appreciate them. Dr. Smriti Bhaduria, Dr. Saman Habib, Dr. S.K. Rath, Dr. Amar Nazar, Dr. B.N. Singh, Dr. Anil Gaikwad, Dr. Anand Kulkarni, Dr. Sanjeev Yadav, Mr. Bhaskar, COA, Mr. Dinesh Kumar, FAO, and Mr. Nilambuj Prasad, SPO, and many others who have come together to make this program happen today. Thank you very much, and I look forward to Professor Talwar's talk. Thank you.
Thank you very much, ma'am, for such inspirational words. Now, please join us and welcome Dr. G.P. Talwar for his Foundation Day lecture.
Well, though 96, I can still do it. Let me first express my extreme pleasure to have come on a day which happens to be also the Foundation Day, 81st Foundation Day of the CSIR. And what is more, to also meet Dr. Nityanand and Dr. Kamboj, of course, over and above you, Dr. Radha Rangarajan, to, for the first time I have had the occasion to shake hands with you. And may you have a very successful tenure as the new director here. This place is very special for many reasons. UP or Abad, Chawaji, kursi laye. No, I'm still going, and let me and I'm engaged in active research still. My current interest is in immunotherapy of cancers of category 2, given up cancers, which are drug resistant and which there is nothing that we can do for the time being. There is no drug against them, nor can surgery be done at the advanced stage. So that is my current interest. But what I'm going to speak to you is on something which has been already indicated, and which are products which have led, the research has gone to the end. And what is my end is not just the finishing of your research and trials, etc., but also its passage to industry so that it is available to the public. So I'm going to speak to you. You see, this long road has also stepwise to go. Even if you have working on a mission to develop a certain product, you have to conduct basic research on it to begin with. And that basic research leads to a lead that, well, maybe something here which could be of utility for the purpose that we are seeking. And then, you have to go through so many approvals at every step, which, by the way, waste so much time. And if at all something could be done to reduce those months and years of approvals from DGHS, from this and from that, etc., it would lead to a much better consequence of what Indians can do for their problems. Anyway, so we have to go through them and go through the usual bureaucracy of ethics committees and toxicology and this and that. But anyway, this first slide summarizes the steps that one has to take to reach somewhere. You have to conduct phase 1, phase 2, phase 3 trials, and before you can say that the product is suitable for industry. And then once the industry you find, and by the way, the industry also is cautious. They don't just accept every product you offer. They also see whether there is any worth in it for profit. And thereafter, it goes to public. So therefore, it's a long road. But, well, nothing gets done without taking a road. Now, we have made some products, and at the time that this slide was made, there were 3 products which had been licensed to industry. So they completed the road, so to say. Oh my. Now, the first product that I am going to speak about is the one which Dr. Kamboj, as the Karta Dharta of Birak, was responsible for giving to an industry in Kerala. It was licensing through Dr. Kamboj, and hence I have included it for presentation. Now, Basant is a polyherbal product. It has curcumin, haldi jo hum khate hain, it is wonderful. Its properties are, you can't imagine how many good properties it has. Amla, of course, we are familiar with. Neem grows in every house, including ours, and so it was easy to collect the leaves, and aloe vera. So this is the composition of Basant. And then, the thing that we tested it for was its action against the many microbes. And obviously, the first was to pick up gonorrhea. And what was amazing is that all strains that we could lay hands on of gonorrhea, including those resistant to drugs, to antibiotics, penicillin, tetracycline, and other ciprofloxacin, etc., they were all sensitive to Basant. And then it was also active against Candida. Candida again has several strains, and all of them were sensitive to Basant. Basant it has also inhibitory action on Chlamydia trachomatis, and then HIV is a problem, or was a problem at the time that we were working on it. What was amazing is that Basant was effective against both CCR5 and CXCR4 tropic HIV1. Normally we know that the drugs are limited in terms of their action on HIV. This particular Basant, just a polyherbal formulation, reacts against all strains of HIV tested so far. And you see what is further amazing is that it can act at the early stage of the disease also, leave aside the. So another very interesting observation was human papilloma virus is the causative factor. It's again, present in the vagina of the women, and it is the virus which causes the, a tumor, and it's difficult to treat. What is amazing is that our formulation acted not only on the isolated HPV, but even HPV which is within the cell of the vagina, and it eliminates that first and then kills the virus. So it's a very interesting property. And when at NIH they tested it, they said, amazing, what have you found? Next slide, please. Then, after having found this, we even conducted a clinical trial, and that was done in Aligarh Medical College, and with Rajshri Sharma, who is the professor of gynecology at that institute. And what we found was, next slide, that all the 11 cases that they tested, every case, 11 out of 11 responded to Basant, and while they were all positive for HPV, but they all became negative, 1 to 11. So it is indeed something which is difficult to say that it is we have made this or formulated it. So it's a very interesting property of Basant to even act on the HPV and therefore prevent a tumor which HPV causes. Next slide. Now the very large number of women at Ames we read, and it was also investigated, that 30 to 40% of women have vaginosis. It's it's a common phenomena amongst women in India, maybe elsewhere also. And what we found was that Basant plus Pro-Veg Health, these are the probiotics that we found earlier, the combination of those clears 95% of patients from recurring episodes of vaginosis. So this is the highest reported in the world literature so far. So indeed, I mean, while it looks, kya choti moti cheezein hain, but they are quite formidable in terms of their properties. This is how a typical case is. On the left side is a woman with the abnormal vaginal discharge, and after using Basant, look how it clears completely. And then here is the clue cells, which after treatment are nowhere visible. So they are also cleared. Next slide. You see the fishy odor, which was also there, and it is in very large number of 40%, 30% of women, all women sitting and doing wherever. At Ames itself, which is a medical institute, there are women who are victim of this process, this vaginosis, and all these symptoms, not only to look at the vagina, but also the fishy odor and the pH of the vagina, all that is restored to what should be the normal situation of vagina. So it's a very, very interesting product which is useful for all this purpose. So here I am ending this story on Basant. It's a marvelous formulation, and it is through Dr. Kamboj that it has been passed on to Mr. Bifa Pharma in Kerala, and it's available to the people not only in India but to the world at large. And let me tell you, Bifa tells us that there is a greater demand for it from abroad than from India, so whatever be the reason. Now I am going to pass on to something which was referred and which I am very proud of. The story is that in 1970, a team of WHO came to meet me in the biochemistry department of the All India Institute of Medical Sciences. They asked me to take over the headship of a research and training center in immunology that they wanted to establish for India and Southeast Asia. I was reluctant. My work on mechanism of action of growth promoting and developmental hormones was going very well, and why shift? But they told me, look, India has world's largest number of lepers. Do you expect Americans to come and solve your problem? Hearing that, I was so ashamed to learn that we have the largest number of lepers in the world. So I immediately signed and took over the headship. And then, question was, first thing was I knew nothing about leprosy. To learn what leprosy is, why people get leprosy. That was the first thing to work on. And we discovered that there is the inability, those who get the disease have the inability to raise immune response against a certain protein of the M. leprae. So therefore, having learned that, the next question was, then can we do something to overcome it, to sort of empower those who are incompetent to resist M. leprae to become competent to resist so. So that is the beginning of the whole story, that and then, to do that, we obviously, whatever you find has to be something which couldn't be used on a large scale. And therefore, we looked for a mycobacteria which could be immunologically competent to do what we are expecting to resist the disease. So you see, we collected a large number of non-pathogenic mycobacteria and tested each one of them for a possible reactivity with the leprosy patients. Leprosy is also a spectral disease. It has a tuberculosis and lepromatous cell. So you see, we had to test a large number of mycobacteria to see whether they are reactive with immune cells or with the lymphocytes of the leprosy patients. And search for an immunologically cross-reactive mycobacteria which elicits immune response from not only tuberculoid leprosy patients but also from lepromatous leprosy patients, the other extreme. And ultimately, we succeeded. and I'm very proud. This is a beautiful bacteria. This bacteria has been genetically analyzed. Its base sequence is known, and as there is none which is described so far in the literature, so it's a new mycobacteria which we discovered, and it's been, you will see it was gene sequence was done, and also this is the bacteria. It's a beautiful bacteria, and it has so many good properties that you will be astonished. So it is named now as Mycobacterium indicus because from India. Pran is my first family name, and NII is the institute from where the trials were conducted, National Institute of Immunology. So that is what it is. It's a fast-growing mycobacteria. Originally, when we investigated it, it was captioned as MW amongst the many bacteria that we were working on. And then you see after it seemed to be the right one for a possible vaccine, we named, but originally it was MW during the investigation stage. And the key antigens of this bacteria were heat stable. So we could kill it and still it would work. So that was the useful property, because then we can make a heat stable vaccine. And that will simplify the toxicology studies. To have a live bacteria and a killed bacteria, the toxicology is of a different nature. So, Chaloji, next. Now, as I told you that the gene sequence of this bacteria has been determined, and that was done by Professor Hasnan, Akhilesh Tyagi and Anil Tyagi's laboratories. That wasn't done by me. And this is the gene sequence of this mycobacteria. And then we determined that it's a ancestor of not only M. leprae, but also of Mycobacterium tuberculosis and avium. So it was named Mycobacterium indicus prani. And it has gone through the whole chain, and it has even been passed on to industry, to Cadila Healthcare, and this is the way it is being marketed as EmuVac. Now let me show you what it does. If you to do all these trials, you cannot cut off the drugs. You have to use them. But you can have two groups, one which receive the drugs, and in the other case you put the additive which is you consider is workable. So that's the way the trials are conducted. And what we could see was that there is to the patients in which we gave also MW in addition to the drugs, their bacterial clearance was faster and they recovered much earlier. Leprosy takes long time for treatment, but in this case, when you combine the MW, it was within 1 year, within 10 months, they were all right, and the extreme cases. And they cleared the granulomas and all. What is interesting is that even the side effects of using the vaccine were reduced and the drugs were reduced. So, this is how it acts. See a case that it's a lepromatous leprosy case to begin with, initial. After 4 doses of the drugs and the vaccine, this is what she becomes. Then after 1 year, look what she becomes. Oh yeah. See, she clears up completely. You cannot even imagine that she ever had leprosy. Drugs have never done that. It is only this vaccine which can clear a leprosy, very ugly case to something which is totally normal, because nobody can imagine that she is a leprosy case. Same here. Look. Here, look all the things in the back, and look at the person after he has been cleared by the drugs and the vaccine. Nothing, no blemishes at all on the skin. See, something which drugs cannot do, this vaccine does. It clears completely the very ugly. See, he was from Nepal, and he touched my feet. कि जी आपने तो मुझे आदमी बना दिया. You know, as if he thinks he has taken a new birth. See, all these. Every case clears completely the ugly blemishes. So therefore, it's a very unique vaccine for leprosy, which not only does the patient is cured in a shorter time, but what is further impressive is that it completely does away with the very ugly symptoms that leprosy patients carry. So all these are gone. Now you see what was again amazing was that it was also active against tuberculosis. We never made against tuberculosis, but perhaps there are cross-reactive antigens, and it also is effective against areas. You see the, oh, sorry. Anyway, oh, sorry. So you see both the spleen and lungs, it's cleared. And here are a trial which has been carried out in category 2 with tuberculosis patients which are difficult to treat with drugs. And there is one group which received the usual drugs plus the MW vaccine, and 48 out of 49 patients receiving the MW in addition to drugs were cleared. In in 97.96 rate of clearance, and then with MDT, which is the multi-drug treatment, normal treatment, only 21 out of 27. So even the number of patients which are difficult to treat in the category 2 tuberculosis, if you add MW along with the drugs, the rate of cure is much better, nearly 100%. What is further amazing is that the relapse rate, the relapse rate at 6 months, 12 months, and 24 months, with drugs alone, it is the dark, you see, these are the drugs alone. And then where the MW has been added, it's the light blue. So relapse rate is much less than what happens with drugs alone, given the normal drugs. So it's a very important addition to tuberculosis treatment. And the National Institute of Tuberculosis in Delhi uses it now for their patients. now I'm telling you something which not many of us expected at all. Why should it be tested against the anogenital warts? We never made it for that. But all these things have been done by others. This is the Sumesh Gupta, a professor at the All India Institute of Medical Sciences. He receives these patients with anogenital warts, and they are so difficult to treat that he didn't know what to do. Then on his own, he said, well, let me use your leprosy vaccine. I mean, there was no logic in it, straightforward, but he tried it just as nothing is working in his hands otherwise. So he gave, and see what happens. See what happens? Clearance, the foot, and given the anogenital warts. See, completely cured. Sumesh Gupta at the All India Institute of Medical Sciences uses it for anogenital warts. This is totally unimaginable, but this vaccine, MW, which is now Mycobacterium indicus pranae, acts on these two. Sumesh Gupta is his name, and 9 out of 10 patients completed, and in 8 patients there was complete clearance. It, to him, is historical, so to say. So these are all his results, which are published, by the way. And at Indian Institute of Science, Bangalore, Dipankar Nandi has tested it against myeloma. And see what happens. The third thing, you see normally the tumor growing, and then with MIP you have hardly any tumor which grows. So it acts against the tumors also. So to sum up then, it's a marvelous vaccine. Even for me, it never expected that we have developed this sort of story, but there it is. And then now of course its gene sequence is determined, it has been named, and it has many properties. The way I interpret it is that it's a potent invigorator of immune responses. So not only against leprosy and tuberculosis, but even anything immune apparatus of the body is upgraded, and it does marvels in many situations where the drugs don't act or difficult to treat. Credit must be given to those who really worked. I was only the guide. So these are all my students and co-workers, staff, who contributed in various ways to various aspects of what I've presented to you. And maybe we should give them credit for all that they have done. Thank you very much. And I'll be willing to respond to any question and query that anybody has. Feel free to pose any question if you have any. Otherwise, I'll take leave, and thank you very much. इसको बंद कर दो भाई. बंद कर दो इसको.
Thank you, sir, for such interesting stories for sharing with us. I would now request our director to kindly felicitate our chief guest.
You are welcome to ask anything, and you are welcome to ask for the lift if you like to use it for something. This also, not only what is inside, but also this, and I'll wear it.
Okay.
Thank you, ma'am. Thank you, sir. It's now time, please sit. Okay, it's now time to announce the Dr. Mridula Kamboj Award for Drugs, Diagnostic Vaccines and Related Basic Research 2021. The group that has been awarded has worked on the technology with the title A Novel Orally Active Fracture Healing Drug Candidate S007-1500 from lead identification to phase 1 human clinical trial. And the group includes the principal innovators, Dr. Atul Goyal and Dr. Divya Singh. Please come on the stage. And the team includes Ms. Pallavi, Dr. Ashutosh Raghuvanshi, Dr. Amit Kumar, Mr. Amardeep, Mr. Krishna Bhan Singh, Ms. Sonu, Ms. Reena Rai, Ms. Kriti Sharma, Mr. Alok Tripathi, Mr. Ravi Prakash, Mr. A.P. Dwivedi, Ms. Dr. Rabi, Dr. Vivek Bhosle, Dr. S.K. Rath, Dr. Sharad Sharma, and Dr. Nivedya Chattopadhyay. Please come on the stage and receive the award from our chief guest, Dr. G.P. Talwar.
Congratulations, sir. Thank you.
Congratulations to the team. And thank you, ma'am. Thank you, sir. Yes, ma'am. On 26th September 2022, CDRI observed it as CSIR Open Day. And on this occasion, various school competitions were held, and it is now time to announce the awardees for these competitions. The first competition was Science Model. The third position goes to Nishita Srivastava from KV Rai Bareli. Although these schools are not available right now, we are just announcing the awards and they will be handed over to the schools. The second award for scientific model is Lakshya Dwivedi from KV Unnao. And the first award goes to Rakshit Kumar Singh, Anurag Debi, and Amrit Mishra from APS Nehru Road. Please, although they are not available here, let's clap for them. Next competition was quiz. The third position goes to Harshi Kumar and Virendra Kumar from JNV Lucknow. Second position to Palak Yadav and Shikhar Verma from KV Unnao. First position goes to Anirudh Yadav and Himanshu Singh from APS Nehru Road. Then there was a painting competition. It was held in two groups. First group was from 9th and 10th standard, and second group was 11th and 12th standard. So the first group, 9th and 10th standard painting competition was held, and second award goes to Tehzeeb Qureshi from Angel Carmel Inter College, and first position again to Angel Carmel Inter College, Pragati Sahu. The second group, awardees of the second group are third position Sneha Singh, JNV Lucknow, second position Vivek Kumar from KV Rai Bareli, and first position Ritika Saroj from APS Nehru Road. And now I would now request our director to give the or honor the meritorious awards of CSIR-CDRI employees. These are the students of the wards of our CDRI staff who have scored more than 90% in at least 3 science subjects, and at least one science subject they have scored 100%. The first name is Miss Ishani Sharma, daughter of Mr. Ashok Kumar Sharma, STO MPC Division. Ishani. Okay. She has scored more than 90% in physics, chemistry, and biology. Second, Ms. Madhushnuhi Panda, daughter of Dr. Gautam Panda, Chief Scientist, MPC Division. She has scored more than 90% in mathematics, physics, chemistry, and computer science 100%.
Okay, please receive it on his
Mr. Anant Gonyal, son of Shri H.M. Gonyal, PTO Department SEV. He has scored more than 90% in physics and chemistry and 100% in mathematics. Mr. Pranjal Dwivedi, son of Shri A.P. Dwivedi, STO MPC Division. He has scored more than 90% in mathematics, chemistry, and computer science. The cash prize for the same will be wired to them. It is now time to announce the awardees of Hindi Pakhwada held from 14th to 29th September 2022.
धन्यवाद डॉक्टर सोनिया वर्मा। इस सभा में उपस्थित सभी लोगों को मेरा सादर नमस्कार। जैसा कि मैम ने अभी आपको बताया कि हिंदी पखवाड़े का आयोजन हर वर्ष हमारे संस्थान में किया जाता है, उसी तरह इस वर्ष भी हमारे संस्थान में हिंदी पखवाड़े का आयोजन दिनांक 14 सितंबर से 29 सितंबर को किया गया। हिंदी पखवाड़े के अंतर्गत समय समय पर विभिन्न प्रतियोगिताओं का आयोजन किया गया। इसी कड़ी में सर्वप्रथम जिस प्रतियोगिता का आयोजन किया गया वह थी हिंदी अनुवाद प्रतियोगिता। मैं डायरेक्टर मैम से अनुरोध करूंगी कि वे प्रतियोगिता के विजेताओं को सम्मानित कर उनका उत्साहवर्धन करें। सर्वप्रथम मैं बुलाना चाहूँगी श्री कुमार सौरभ जी को, जिनको कि हिंदी अनुवाद प्रतियोगिता में तृतीय पुरस्कार प्राप्त हुआ है। मैं चाहूँगी जोरदार तालियों से इनका स्वागत किया जाए। सौरभ कुमार जी अगर सभा में उपस्थित हैं तो कृपया कर मंच पे आने का कष्ट करें। द्वितीय पुरस्कार जिसे प्राप्त हुआ है वह है श्री रमेश चंद्र गुप्त जी। मैं सर से अनुरोध करूंगी कि मंच पर आकर अपना पुरस्कार ग्रहण करें। प्रथम पुरस्कार के लिए मैं बुलाना चाहूँगी श्री प्रियंका गुप्ता जी को। शायद वो अभी यहाँ पे उपस्थित नहीं है। नेक्स्ट जो हमारे हिंदी टिप्पण एवं आलेखन प्रतियोगिता आयोजित की गई थी, उसके प्रतियोगिता के विजेता हैं श्री दीपक गुप्ता, जिन्हें तृतीय पुरस्कार प्राप्त हुआ है। द्वितीय पुरस्कार के लिए मैं श्री अनूप ठाकुर जी को बुलाना चाहूँगी। और प्रथम पुरस्कार जाता है श्री विभास कुमार जी को। हिंदी आशुभाषण प्रतियोगिता के लिए मैं दोबारा विभास सर से अनुरोध करूंगी कि वो आप तृतीय पुरस्कार ग्रहण करें। द्वितीय पुरस्कार के लिए श्री पंकज कुमार जी को मैं आमंत्रित करना चाहूँगी। प्रथम पुरस्कार जाता है सुश्री प्रतीक्षा तिवारी को। इसी कड़ी में पूरे वर्ष हिंदी में टिप्पण एवं आलेखन प्रोत्साहन पुरस्कार के लिए मैं तृतीय पुरस्कार के लिए बुलाना चाहूँगी श्री अनिल कुमार जी को। सुश्री निदा प्रवीण जी। श्री विभास कुमार जी, अजय शुक्ला जी, श्री जय सिंह। द्वितीय पुरस्कार के लिए मैं बुलाऊंगी महेश कुमार जी को। सुश्री अंजलि सिंह, श्री अनूप ठाकुर जी, प्रथम पुरस्कार के लिए मैं बुलाऊंगी श्री कांत जी को, सामान्य अनुभाग। हाँ, मेरे से गलती हो गई, ऋषि कांत जी को। इसके बाद दीपक गुप्ता जी। वोट ऑफ थैंक्स के लिए मैं डॉक्टर सोनिया वर्मा को दोबारा एक बार मंच पे आमंत्रित करना चाहूँगी।
Before the vote of thanks, it's now the time to felicitate our colleagues who have superannuated in previous 2 years, and also those who have completed 25 years of their service. And let's begin with those who were superannuated from September 2020 to August 2021. Please come, I request them to please come on the stage and receive our good wishes. Shri Om Prakash Verma, Shri Ram Avatar, श्री राम समुच।
2020 के हैं, मेरा 20 में नाम है।
हाँ जी, आपका नाम। हाँ हाँ हाँ, अभी आपका नाम, हाँ ठीक है, ओके। श्री ओम प्रकाश वर्मा, श्री मधुसूदन वर्मा, प्लीज यहाँ वेट कीजिए सर, प्लीज वेट कीजिए। क्या करेंगे? श्री के सिंह, श्री राजेंद्र कुमार श्रीवास्तव, श्री संजय कुमार, श्री रमेश कुंवर, नहीं people they have not come. रमेश जी, रमेश, श्री रमेश कुंवर, श्री विनोद कुमार मौर्य, श्री प्रमोद कुमार, विनोद जी, श्री विनोद कुमार मौर्य, श्री हरि दुआरी, ओके, श्री प्रमोद कुमार, श्री हरिद्वारी, श्रीमती रीना बिसारिया, श्री कृष्ण कुमार, श्रीमती सावित्री देवी, डॉक्टर नीलू सिंह, डॉक्टर दयाशंकर उपाध्याय, श्री सुनील कुमार, ओके, श्रीमती रीना बिसारिया जी के हस्बैंड प्लीज स्टेज पर आए। वो आपको थैंक यू बोलना चाहती है आपके सपोर्ट के लिए। कहाँ पे है? ओके, डॉक्टर दयाशंकर उपाध्याय, थैंक यू सर। श्री सुनील कुमार, श्री हरि प्रसाद, श्री अरुण शर्मा, सुश्री विजय अशर, श्री दिलीप कुमार, श्री जेपी सती, Shri Sandeep Sen Gupta. Shrimati Nandita Pandey. Shri Jamil Beg. Shrimati Manju Yadav. बहुत लोग नहीं आ रहे हैं ना।
नंदिता जमील बेग। Whoever is not here, you say they are not here so that they know.
Okay.
You scan the audience. If somebody is moving, stop and let them
ठीक
come.
है ठीक है. Shrimati Manju Yadav. Not present. Dr. B. Mehti. Shri Rampal. Shri Hira Ballabh Neolia. हाँ. Shri Krishna Kumar. Now we'll move to our colleagues who are superannuated from October 2021 to August 2022. Shri Ashok Pandey, Shri Kamal Singh, Shrimati Tehseen Tilat, Shri Avdesh Narayan Dikshit, नहीं है। श्री करुणेश राय, श्री सत्यनारायण, श्री अरुण वढेरा, श्री वेद प्रकाश मिश्रा, ओके, नो, ठीक है। कंचन बालाजी वडेरा जी का कलेक्ट करेंगे गुड विशेस। श्री वेद प्रकाश मिश्रा, श्री अरुण कुमार श्रीवास्तव, श्री राजेंद्र, श्री VBL श्रीवास्तव, डॉक्टर नीना गोयल, श्री प्रेम प्रकाश, श्री दिलीप कुमार खरे, डॉक्टर गोपाल गुप्ता, श्रीमती मधु चतुर्वेदी, श्री सुरेश कुमार वर्मा, श्री नंद पाल यादव, श्री नवीन प्रकाश मिश्रा, श्री भारती भूषण,
Mishra.
श्री भारती भूषण, श्री पंकज बहादुर, श्री सुखदेव प्रसाद, श्री महेंद्र कुमार, श्री राजू अरोड़ा, श्री रामजीत, It is now time to felicitate those of our colleagues who have completed 25 years in 2021. Shri Narendra Kumar, Shri Anil Kumar Verma, Shri Ram Lakhan, Shri Dinesh Kumar, Shri Pradeep Tikri, Shrimati Manju, Shri Mahesh Chandra Tiwari, Shri Satish Chandra Yadav, Shri Satish Chandra Yadav, Shri Ram Chandra Maurya, Shri Rajesh Kumar Verma. Now the CDRI staff who has completed 25 years in 2022. Shri Mohit Kumar Shukla. Shri Ram Karan Harijan. Mohammad Irfan. Mohammad Irfan. Shri Gajendra Prasad Tripathi. Mahesh Kumar. Thank you, ma'am. You may join us in the audience. Thank you very much. I would now like to request the chairperson of our organizing committee, Dr. Smriti, for the vote of thanks.
Thank you, Sonia. A very good afternoon to all of you. Now that we are at the culmination of this 81st CSIR Foundation Day celebration, I stand here gladly to express my vote of thanks to each and every individual who was part of this journey, which was a learning experience for all of us. On behalf of entire CSIR-CDRI family, I express my deepest gratitude to Professor G.P. Talwar for accepting our invitation and gracing the occasion. Thank you, sir. Your insights and your ideas have greatly inspired and motivated us. Today, we are also joined by our respected mentors, Dr. Nityanand and Dr. V.P. Kamboj. Our heartfelt thanks go to them for being here with us today, despite the busy schedule. Their presence always encourages us and motivates us to reach ahead. Our Honorable Director, Dr. Radha Rangarajan, who joined CSIR family very recently, she guided us at every step for putting the entire program together. Not only was she always there when we needed her, she made it sure that function is celebrated with enthusiasm. Thank you, ma'am, very much for your guidance and constant support. I also take this opportunity to thank then Acting Director, Dr. Nabedia Chattopadhyay, for entrusting me with this responsibility of organizing such an important event. Thank you, sir, for the opportunity and for the valuable learning that came with it. A function like this essentially requires concerted efforts of multiple departments and committees with constant improvisations at every step. I offer my deepest gratitude to all the conveners of the subcommittee, respective members, departmental heads, and staff members for managing multiple and diverse aspects of this program throughout with immense dedication. After a hiatus of 2 years, we once again opened our campus for students of nearby schools and colleges. This year, we had more than 500 students visit us on open day. It indeed was a wonderful experience to see these young minds interacting with our faculty, staff, and students. I thank all my senior colleagues for enormous support that they offered throughout the planning and execution phase of the program. And this vote of thanks would be incomplete without thanking CDRI students, both PhD scholars and project fellows, who rallied behind us with full dedication to make this Open Day event a grand success. And last but not the least, I would like to thank each and every individual who is present here in the audience for being here with us this morning. Thank you all.
Thanks, Dr. Smriti. I would also like to thank Shraddha for helping me in announcing the awards. Now I've been asked to inform all the individuals who have received the felicitation to kindly give us your receiving signatures backstage. Now it is time for the national anthem. Please rise.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you. Kindly join us for the high tea at back of the auditorium.